Hello, everyone. Welcome, and thank you for attending the WormBase Webinar series. My name is Karen Uke. I'm a curator at WormBase and an editor for the MicroPublication Biology Journal, which is an offshoot project of WormBase and the subject of today's webinar. Before we get started, a few quick housekeeping notes. With respect to Zoom, your line has been muted and video disabled in the interest of a smooth presentation. Should you have a question, please either send it in the chat or raise your hand and a moderator will get to you. We will be taking time after the presentation to answer any questions you've posted during the talk. So please stay online after our speaker is done. Okay, so with that, let's begin. Today's webinar focuses on the MicroPublication project. MicroPublication is dedicated to the process of simplifying scientific publication and depositing that published research in downstream community knowledge bases. You can visit us at micropublication.org or follow us on Twitter at micropub7n. Those will be posted in the chat. And today you will be hearing from Tim Shadle from Washington University, St. Louis. Tim is a long-standing C. elegans researcher and a founding member of the MicroPublication Project. Tim is also a C. elegans science officer for micropublication biology. So without further ado, it's a great pleasure to introduce Tim Shadle. Take it away,
Hi,
Tim.
everyone. Okay, let me share my screen. Okay, can everybody see everything?
Yes.
Yes.
Good, good. All right, so I'll take it away. So MicroPublications, is a novel publication platform, and we started it really with two major goals. And the first goal is to increase the entry of research findings into findings and methods into the public domain. And these would be findings that are of value to the community, and some of these might not otherwise make it into the public domain. Importantly, publication gives researchers credit for their work as a publication on their CV, and of course with PubMed indexing. But furthermore, publication fulfills agreements researchers make with funding agencies and of course the taxpayers to disseminate their findings. Now, the second goal is to facilitate incorporation of new findings and information into scientific databases like WormBase. So databases are of real value to researchers and facilitators of research because they aggregate data and they make it easier for researchers to find, and this speeds up the scientific enterprise. However, databases are of most value when they're comprehensive. And NIH is constantly cutting funding for databases. And so therefore, other methods really need to be moved forward to try to make and maintain database comprehensivity. And so what is a micropublication? Well, it's a peer-reviewed scholarly report of findings from a single experiment. However, the term single experiment, isn't really a single experiment because it involves controls, biological replicates, and there may well be actually, multiple phenotypic or biological readouts that are obtained from that experiment. So it isn't truly a single experiment. Typically, micropublications have a single figure or a table that's associated with that. And the figure, typically has multiple panels that are showing various different data types, explaining the experiment, giving a summary model, for example, and to really allow the authors to really say what they and present what they need to, we allow the figure to be up to 2 pages, PDF pages in length. This also allows it to be large enough to be visible in a single in a single or at 100% in the PDF. Now where relevant, a separate data table is important. And this is because data tables are machine readable, which will facilitate getting the data into databases. Okay, so in terms of the text, it's really brief and to the point, and typically has sufficient background for the scientific community to understand what is the question or issue, and then how it will be approached. And then a description of the experimental details and the findings and conclusions. Also, it has methods and reagents, and the methods should be in sufficient detail to allow it to be repeated by others or referred to other papers where the methods are described in sufficient detail. And then, of course, it has the standard references, acknowledgements, funding, author contributions. Now, micropublications really are scholarly presentations of research, and they need to meet the standards of the field. And one of them, of course, is that they need to be well written and comprehensible to the audience. And we have, on occasion, editorially rejected papers, really due to poor writing. And so these need to be, well written, documents. They also need to be scientifically sound and rigorous. For example, there needs to be biological replicates. In the case of quantitative data, there needs to be statistical analysis. And of course, the controls, control experiments, must show results that are compatible with the literature. Okay, so the categories of micropublications are, new findings. This also includes new reagents or new methods. Findings that were not previously shown, and this would include in other publications, things that are indicated as unpublished results, for example. We also publish negative results. And in fact, NIH recently inquired to us, how many negative results have we published, because they feel that this is really part and parcel of rigorous science. And then we also publish replications that are successful and replications that are unsuccessful. And we also publish community validations. So now I'm going to just briefly go through a few examples of micropublications, and you, of course, should really go into the journal and look through and see, the breadth and the depth of publications that we have. So one example here is, on the left here is protein localization for extracellular matrix proteins. On the right is a really very interesting paper showing that the HSLN neuron, which is of course well known for egg laying, is also functions in the defecation motor program. On the left here is a micropublication showing that the FAT 5, 6, and 7 genes function in body size. And on the right is a new deletion allele, probable null for the SPCH3 gene. On the right here is an expansion of the SAPTRAP genome engineering repertoire. And on the right is a novel protein engineering method to use rapamycin to get induced dimerization. So here's a list of the data types that are currently being published or that currently have been published. But we certainly welcome any new other data types that have not currently been published. So, an important thing about micropublications, given that they're really single experimental results, they are quick and easy to review. And for example, are these, excuse me, Is the work experimentally sound? Does the data support the conclusions? Is there sufficient information to allow replication by others? And are the findings of potential value to the community? And the peer reviewers can either be anonymous, or that they can get credit for their peer review if they indicate that they're interested in this, and that's at the end of the article. So now, there are a number of outcomes from the peer review. The first is just accepted without modifications, and we have had a few examples of this. There's also accepted with minor modifications, either to the text or the figures, accepted with major modifications to either the text or the figures. Now, in general, it's not expected that authors would need to do new experiments. However, there are exceptions to that. And so here are a couple of exceptions. One would be, for example, say we received a micropublication with a DAF2 allele that reveals a brand new phenotype that has never been observed before. However, the authors did not look for this phenotype with the canonical E1370 DAF2 alleles. And so here we would ask the authors to either repeat the protocol using E1370, or the authors to have an explicit caveat saying that while we found this phenotype for allele XYZ, we don't know whether the canonical E1370 allele shows the phenotype. So the other situation is where there is a missing essential data, for example, a control genotype, or a genotype where the authors are making conclusions. And so here the authors either need to provide the data, and oftentimes they actually have the data and they just didn't include it. But if they don't have the data, either the experiment needs to be done, or the paper will be rejected because there's not sufficient information for the community to judge the soundness of the conclusions. And finally, there's just a rejection. In addition, we also have confidential comments to the editor, and these would be comments such as a worry about plagiarism or inappropriate figure manipulation, or concerns about the data quality in terms of not sort of meeting the standards in the field. So, so far we have published 257 micropublications, and 205 of these are from Worm, and then we've had some others from Fly and Arabidopsis and Saccharomyces, Xenopus, and zebrafish. We are currently rejecting it at a level of about 5%, although a number of articles before they were published went through multiple rounds of revisions to meet our standards. So all publications are now indexed in PubMed, European PMC, Google Scholar, and they receive a citable DOI. And here's an example of PubMed showing the titles for some micropublications. So now I think it's useful to compare and contrast micropublications from sort of standard publications like an article in Genetics or Journal of Cell Biology or Development. And so a standard publication really is a line of investigation, and these take multiple years, and these generate multiple results that lead to multiple figures and tables and multiple supplemental figures and tables. And these lead then to a number of conclusions. And such standard publications really are important for promotion and tenure, and they're important for receiving national competitive grant funding. Micropublications are different, and again, they're now a single experimental findings, and and so one then needs to sort of judge in your repertoire of experimentation that's going on in your labs or experimentation that you're involved, what is suitable for a micropublication versus what's suitable for a standard publication. So here are just some examples that I can go through. One would be a finding that is of value to the community now, but the project is in an early stage and won't really come to fruition for 1 or more years. And this, so for example, would be a result that would be of value to the community. And if you want to start getting cited for that now, or you're worried about being scooped for that result, then you should go ahead and think about micropublication of that. That result can be included in your large publication as a citation showing, yes, we developed this transgene to allow expression, and then you make a citation for it, for example. Another would be useful and concrete findings, but these really are tangential to what you're doing or what the lab is doing, but still could be of use to the community. Another example would be a solid result, but it didn't make it into the relevant publication. And so, if it's a value to value or potential value to the community, it should be considered for a micropublication. Another would be, a student or postdoc has left the lab or will be leaving the lab soon, and it's not clear when that work would be followed up. This is a prime example of something for a micropublication. Another would be a summer undergrad research project, and then this would give credit to the undergrad. This also allows the undergrad to go through the process of well, how do I present this information in a concise but informative way. Another would be a standalone finding that's part of a larger research effort. Maybe you would rather than being a middle author on a large publication, you might prefer to be first author, for example, of a supplemental figure. And additionally, you could imagine that as part of an undergrad course, if you design the course in a way that it could lead to a micropublication, that would be great. And so next, I'm going to show you the classic micropublication from Watson and Crick. And we're hoping that some of our micropublications will have equivalent impact. And so here's the micropublication leadership team. Here's Paul, Daniela, Karen, Todd, and Nick. And so Paul and I are open for questions.
Okay. Are there any questions?
While we wait for the questions to come in, I have a question. I'm Wen Chen from WormBase. I just curious, because this is micropublication biology, but I just wonder because I know in the bioinformatics field, there are many people writing small bioinformatic tools and things like that, and some of them do not get published. So is it possible to have a micropublication category for these kind of softwares? People can publish their websites and then show where the GitHub repositories are, so that in the future, if anyone wants to use this or continue to develop this software, they don't have to reinvent the wheel. I particularly ask this because in WormBase, every time we migrate to a new platform, we lost something. It's just difficult to go back and dig out those softwares. So do you have any plan?
Yeah. So Wen, that's a great question, and I'm going to give you sort of two-part answer to that. So the first part is, if the software is clearly related, or being applied to worm biology, then that can be part of our current platform. However, our model for micropublication biology, micropublications in general, is really bottom-up, publishing rather than top-down publishing. So top-down publishing is Elsevier, for example, or Nature, and they decide, we're really interested in geology, we're going to start a journal on geology. But that's not the model for micropublication. There really needs to be a community of scientists, of PIs, that want to micropublish their work, and they want to be involved in the peer review process. And then a micropublication journal can spring from that. But there really needs to be a community of researchers. And typically in the biological realm, there's a database that's actually organized around that. And so, for example, the Drosophila publications that I mentioned, of course, there's fly-based information, and so a lot of the biology goes in there. So in terms of bioinformatics, I think if it's clearly related and can be connected to the C. elegans part, it could be submitted as is. Other kinds of things, I think there would need to be a bioinformatic, specialty.
Yeah, I just wonder, maybe you can have, because for micropublication, my understanding is you have this web form, something standard, makes it easy to submit and publish. So because in WormBase, my understanding is we're going to migrate it into the Alliance of Genome Resource. So I expect quite a few of our current tools will retire, but although they all have repositories, but without publishing them, I just feel it would be difficult for the future people to find how to get these tools reinstalled or continue to publish them. So it would be really nice if we have something standard for people to submit these kind of softwares.
Okay, so let me also turn over the mic to Daniela, who will handle all the questions. So please continue to post questions in the chat or raise your hand and we'll get to you.
Thanks, Karen. Yeah, we're getting some questions right now. So the first one was from Jacob. Jacob asks, he's new to WORM, what is a community validation? And I think it was commodity validation that Tim was referring to in his presentation. So Tim, do you want to explain that?
Yeah, I mean, so for example, Nemometrics had a new way of looking at a phenotype using one of their commodities, and so they, benchmarked the assays that could be done with their commodity.
Thank you, Tim. So the next question is if there is an overall page limit, and that's from Catcher Salazar.
There is not an overall page limit. However, I think, given essentially you're really presenting, a single experiment, and the associated things that go with it, there should be a premium on being concise, and not waxing too poetically. We do also have a platform called integrations, where more than one experiment can be done, where each experiment has its own micropublications, and then they're integrated in with a sort of a summary discussion like micropublication. So there isn't any true length limit. And so, for example, if somebody is presenting a new method, the details of that, method should be explained, and that can, and that can be, as long as necessary given the complexities. I would say our typical micropublication, basic description part, has between, on the order of 500 to 750 words. That's the typical ones.
Thank you, Tim. The next question is, who can review micropublication, and is there a sign up? I can first say that there is a sign up. If you go to micropublication.org and click on submit a micropublication, at the bottom of that page there is a link to a Google form, so anybody can sign up. And I let Tim address on who is our idea reviewer and who can review micropublications.
Yeah, so I think typically micropublications are reviewed by principal investigators. However, if a principal investigator says, oh, a postdoc in my lab, or a staff scientist in my lab is an obvious expert and can evaluate it, that's a situation that we've dealt with. And we've certainly also dealt with situations where a senior graduate student has sort of co-reviewed the article with the PI, or the PI sort of signed off on the review.
Great, thank you, Tim. So Adria is asking, sometimes unraveling new info in previous articles, let's say micropublication, can these cause problems for journals not accepting already published data as not being novel anymore?
Yeah, I mean, I'm not completely sure what you mean for the question. Clearly, if you publish, so for example, I think, if you publish a data figure where you're making a conclusion, you can't sort of republish that, in another article. Even as, figure 2 of a 10-figure article. But you can cite it and say, we previously showed that, gene X promotes cell type Y sulfate, and then you go on from there. Does that sort of help?
I'm checking in the chat if Adria, does that answer your question?
Paul, did you have something to add? It looked like you wanted to.
Yeah, no, I just say, I think, if you're concerned, don't micropublish it, right? When I tell people to write it up as a micropublication and then don't send it in. And then 3 years later, when you realize that other paper didn't get written, you can ship it in, right? But be conservative. We don't want to mess anything up.
Thanks. So the next question is, what is the current average time from submission to publication for micropublication? I think we calculated that, Karen, if I'm not mistaken, it was around 2 weeks, but that varies greatly based on the how fast the authors, if they're asked to do revisions of the paper, are turning in their revision. So we had Gold Cases example in which a micropublication was published the same within a day from acceptance. And then we have—
Including a revision, right? Including revision.
Including revisions. That was pretty spectacular. And then the rest depends basically on the reviewer responsiveness and on the author responsiveness in addressing the reviewer's comments. Paul, Tim, did you want to add on that?
No, that's good.
I mean, we could post a distribution, right? I don't know if we posted.
We should, yeah.
Yeah, I mean, the critical is the median, not the You know, because you could occasionally have one that lasts, you know, they take 3 years to publish because the author sat on it.
All right, moving on. Sanjeev was asking if there is impact factor. Do you want to take this, Paul and Tim?
Paul, you want to take that?
Well, I think, you know, the impact, it's too early to know. I mean, I think you're, you know, I think at one point we're not concerned about that. I think this is something where you want to use individual article level citation, right? So the only thing that's, because there's going to be a range, there's going to be negative results that'll never get cited. So we're okay with that. So I think if you're concerned about impact factor per se, at this time, this is not the place, right?
Right.
But
negative,
it's gonna, you know, it has value. I think there's CV value to it.
yes. And negative results which don't get cited are still useful to the community, because then somebody doesn't go off and try doing that experiment when they know it's going to fail.
Also, it might get included in the database and then used for artificial intelligence, you know, sort of training sets, but it might not get cited because it'll be accumulated in the database. So it'll have good value. But yeah, so I think, you know, the way to think about this is your article going to get cited or not, right? So we're unapologetic here.
Yeah, I just wanted to add that we're also in the process of applying to the Web of Science to be indexing Web of Science, and based on the evaluation, then some journals will be selected for impact factor. So that's another thing to consider.
Well, also the other thing is that that's really important for our citations in micropublication to count, because I think that will actually have a good effect on papers. You know, it'll change the journals that get cited, I think.
Right.
Right. Because if you have an article about, yeah, go on.
No, go on.
Yeah, no, I'm saying if you have an article that, you know, it has one experiment, you can actually cite all the right strains and reagents, right? So you're going to give more credit to the people and papers that deserve credit. So I think that'll help the citations in general.
Thanks, Paul. The next question is still for Paul. Chia Chang asked, Paul, does micropublication plan to publish any mini review or comment type of papers?
So yeah, okay. Yeah, so I'm trying to figure out, we're trying to figure out ways to get micro reviews. And there's a couple of purposes. One is that it can be kind of updated. Okay, the experience with Wormbook, when we were trying to get them revised, it was kind of onerous for people to revise pieces because they're long articles. And if you have a short review, it's easier to update that, right? Because it's more granular. That's one thing. The other thing we're trying to do with pathways, we have an idea of enlisting experts to describe a pathway with the evidence and then write a micro review about that. And that will really help the knowledge bases across the board. So we think this is a good opportunity to rigorously define pathways of all types, for example. Commentary, I don't know if you have ideas. I think we've discussed some sociology and commentaries. We have to just see what comes in. If that answers the question.
Tia Chang, does that answer the question? Yes. So Sanjeev is asking as a follow-up, for these micro reviews, do you need the consent of the PI, or students and postdoc can write it by themselves?
Yeah, so typically articles need the consent of and agreement of the PI.
Thank you. Tim, then we have a question specifically for you from Evandro Fang. So Evandro is asking if it's possible to have a webinar on worm nomenclature, as he reached out to you in the past asking many questions about worm nomenclature.
Yeah, I guess it depends a little bit on how much, you know, demand there is for that, you know, rather than just sort of answering the questions one-on-one.
Yeah. We might think to add one to the WormBase webinar series if there's interest. Catcher Salazar is asking another question. If micropublications that constitute an integration, are they published, they have to be published at the same time, or if they can be published throughout a time period? Paul, do you want to address this question?
So the question is about, say this again, about the time period?
About integrations, if they have to be
right,
published
yeah.
all at the same time.
No, I mean, I think, you know, they don't have to. I mean, we don't have enough cases to see how it works out. So, I mean, it could be a little easier to write if they're together, but no, I think that's one of the advantages of rolling out stories, right? Yeah, so I think it's up to you.
Catcher says, okay, thank you. Yeah, Raymond is saying that we might add a nomenclature webinar as a poll in the webinars. We will do that to see how many people are interested. We'll wait a couple of additional minutes to let more questions come in. In the meanwhile, Paul and Tim, if you want to stress some points or have additional remarks, please.
If you're going to do a nomenclature webinar, I'd get somebody from the Drosophila community to come in and have a little dust up about it. It'd be fun. So one question, okay, one comment about the integrations and publishing as you go. So one concern about micropublication that was brought up is a lot of times the validity of results is tested by follow-up experiments, right? So the advantage of the waiting a long time to publish is if there's some mistake in a clone or a strain or reagent, it gets sorted out. Hopefully it gets sorted out eventually, or is more likely to get sorted out if you wait. And if you published every 2 weeks, you would be publishing things with those mistakes, right? So we believe that replication and reproducibility is better served by micropublication in general, but there are going to be cases where someone published too soon. So I think if you have the integrations and it is better to have some delay in publishing, right? If that's okay, right? Like I just don't, I mean, the absurd would is if you published, as soon as the experiment was done and replicated, sometimes in my experience, I've seen people realize that they held the gel upside down or something was wrong the next day. So you don't want to be too quick, right? We want to be quick, but not too quick. If that makes sense.
Thanks, Paul. We'll wait another minute to allow you to put in more questions for the micropublication team.
Daniela, did you ask the question about community validation? I think someone entered very early.
Yes, we did when. Thank you.
Great.
Okay, Karen, back to you.
Yeah, I'm going to just share my screen for some closing comments. Here again is the contact information if you have more questions about micropublication. Thank you. Probably had a lot of time to look at that. And also, let me just, this concludes the WormBase webinar series episode on micropublication. Thank you for a great overview of the micropublication project, Tim. And thank you everyone for joining. We hope you are all as excited as we are about this project, and I look forward to helping you get your data out to the public through a micropub. While we address the questions you, okay, so that concludes this episode. Hope to see you at future episodes. If you have any questions concerning the webinar series itself, please write to outreach@wormbase.org, and hope to see you on another series, another episode. Okay, bye everyone.
Bye.